the conveyor belt, the boxes, the shredder, the FcRn scanner. You know the system. But what if you could predict exactly how a box moves, its clearance, its volume, its half-life, just by reading the barcode? No test factories. No animal studies. Just the amino acid sequence and an AI that learned the patterns from 118 mAbs. Traditional mAb development takes 12 to 18 months before you can predict human pharmacokinetics. Design the molecule. Produce it. Test it in transgenic mice. Test it in cynomolgus monkeys. Fit a model. Scale to human. Each step costs months and millions. But the amino acid sequence is known at the design stage, before a single molecule is manufactured. If the sequence contains enough information to predict PK, you could screen thousands of candidates in seconds. The researchers compiled a dataset of 118 diverse monoclonal antibodies with measured pharmacokinetic parameters in humans. For each mAb, they extracted two types of features from the amino acid sequence. First, a protein language model, trained on millions of protein sequences, converts each sequence into a numerical embedding that captures evolutionary and structural information. Second, sequence attention mechanisms highlight which residues are most informative. Combined, these features represent each mAb as a numerical vector that an AI can learn from. Here's the key innovation. The neural network doesn't predict PK curves directly. It predicts the four parameters of a two-compartment model: clearance, intercompartmental clearance, central volume, and peripheral volume. Then the two-compartment ODE, the same physics model from our one-model-fits-all video, generates the concentration time curve from those parameters. The loss function compares the physics-generated curve to the observed clinical data and backpropagates the error through both the ODE solver and the neural network. This is a physics-informed neural network. The AI learns biology-consistent parameters, not arbitrary curve shapes. The most fascinating result isn't the accuracy, it's what the AI learned about antibody biology. The sequence regions most predictive of clearance aren't the commander loops that bind the target. They're the framework regions and the Fc domain, exactly the parts responsible for FcRn binding and non-specific interactions. The AI independently discovered what Betts and Avery showed empirically: clearance is an Fc property, not a target property. The sequence features predicting fast clearance correlate with the same charge and hydrophobicity patches that the AC-SINS assay measures. The AI found the biology without being told. 13 mAbs held out entirely from training. The AI had never seen their sequences or their PK data. Fed only the amino acid sequence, the physics-informed neural network predicted clearance within twofold for 10 of 13 mAbs and volume of distribution at steady state within twofold for all 13. The three outliers all had unusual properties: non-standard Fc modifications or high self-association. No animals. No experiments. Just the sequence. Imagine designing 1,000 mAb candidates computationally. In minutes, the AI scores each one for predicted PK, clearance, half-life, therapeutic window. You filter to the top 50. Synthesize only those. Validate the top 10 in TG32 mice. One cynomolgus monkey study for the lead candidate. 90% fewer animal studies. 12 months faster to the clinic. 1,000 times more candidates screened. The AI doesn't replace experiments. It tells you which experiments are worth running. 3 videos. 3 eras. The universal model, one blueprint fits 109 factories. The barcode hack, engineer the Fc, extend the ride. The AI scanner, read the sequence, predict the ride. From conveyor belts to physics-informed neural networks. The future of monoclonal antibody pharmacokinetics.